So today, my name is Thor Rollins. I work at Nelson Labs. I've worked at Nelson for almost 15 years now. And I'm the senior scientist over at Biocompatibility at Nelson Labs. And today, we're going to be talking about something that I do quite a bit, and that is evaluating changes of medical devices. It seems like engineers, specifically engineers, can't stop tinkering with a medical device. It always has to make it better, which is great. But unfortunately, every time that we make a change, we have to evaluate what that change means to the patient. And even though it might better the functionality of the device, we have to evaluate the impact of biocompatibility. So I found this quote that I thought was very appropriate for this type of subject. It says, "Any change, even a change for the better, is always accompanied by drawbacks and discomforts." And so I think I feel the discomforts when you guys make a change more than anybody, but we've learned how to get through those changes without having to redo all of our biocompatibility testing, and that's kind of what we're going to be talking about today. But I want you to remember, and I get this all the time, there are no safe materials. Everybody's like, well, we're just changing it to another polypropylene, or we're just adding titanium, and titanium's safe. Well, I'll give you, if you implant titanium, it's going to be safe. But if you throw a bunch of titanium into the vascular system, it's not going to be safe. Or if you inhale a bunch of titanium particulates, it's not going to be safe. So there are no safe materials, only safe doses, okay? So we need to kind of not think about how the material is used. We need to think about how is it delivered to the body. So that new change, that new material that we're adding, what does that impact have to the body? And also what potential processing we've added to the device to impact it. So what we're really doing is when we're changing the device, we're evaluating the risk of that change. So what is risk? Is the probability of an occurrence of harm and then the severity of that harm, and that's risk. And so with any change of the device, we need to evaluate, does that change increase the severity of harm of the device or the probability of that harm? And that would help us impact or decide if that change is beneficial or not. Everyone's familiar with this flow chart. This is the flow chart of ISO 10993-1, and it's a decision to kind of help evaluate biocompatibility. The same concept holds true for changes. And the reason I wanted to point this flow chart out is I always notice, or down here, the only part it talks about testing is at the very bottom of the flow chart. So many people jump right to just repeating testing or doing testing as the way to evaluate biocompatibility. But the standard actually has us go through so many different options before jumping straight to the testing. And in fact, as you see, there are exit points all along the flow chart that allows us to do an evaluation instead of doing testing. And the same thing holds true to changes. In fact, it's actually easier because most of the time we're not doing a brand new device. We're only evaluating a small impact to an already approved device. So, in my experience, there's 4 general types of changes that I see in medical devices. We have material changes, where we're changing a material in the device, or a manufacturer of the same material. That's the next change, is a manufacturing site, or the company manufacturing that material. A process change, and that includes sterilization. We're changing from gamma to EO, or we're changing an adhesive administration or mold release to something else, or we're changing dimensions. And sometimes dimensions gets overlooked. A lot of people think that a dimension of a medical device may not have a big impact in the body. But when you're looking at hemocompatibility or implantation, the dimensions of the device can have a huge impact. So even though the materials and processes may stay the same, if you're changing the overall dimensions of a medical device and it has implantation or hemocompatibility involved in it, then those changes can have an impact in the biocompatibility, or potentially can. So these are the 4 changes that we generally see with medical devices at Nelson Labs. All 4 of these changes we can evaluate with minimal testing. And I'm going to kind of help you go through a decision process that we use internally to help decide the impact of a change, and then some of the testing that we use to evaluate the potential of that change. This is a table out of ISO 10993-12. It's table 1. And this, for me, is the most useful table for an evaluating change. To give you a little bit of idea about the table, how it works, is this is the table we use to determine extraction ratios. So you give me a device and we extract it, right? We put it into a liquid and we get the chemicals that come off that device. These ratios are dependent on the surface area or the thickness of the device. All that is what the lab uses. You guys don't really need to know that. But the one part that I do want to point out is this plus or minus 10% on the top of the chart. Basically, what that plus or minus 10% allows is for a variance in surface area at the test to be the same. So let me give you an example. Let's say you do a test at Nelson Labs, and we calculate the surface area 60 centimeters squared. You do a test at one of our competitors, and they calculate the surface area 55 or 56 centimeters squared. What this table means is that plus or 10% means those tests can be comparable, okay? Honestly, what it's really telling us is that a small difference in surface area does not mean a big impact in the test solution. So a small change, plus or minus 10%, means small impact in the results of a test. And that's what this chart is telling us in that plus or 10%. So us as labs, we use that to evaluate the impact of a test. We also now use that to impact or evaluate the impact of a change to a medical device. To give you kind of an idea about how the surface area plays a role in my testing, I'm going to give you an example. Right here is a partial knee implant. It's a very common medical device that we see a lot come in for testing. And we have, per this chart here, we have two ways we can test it, either surface area or weight, okay? So by weight, this device weighs 93.9 grams. So per that chart I showed you, we would extract in 0.2 grams per mil. This would give us 468.5 mils of extraction fluid for this knee, okay? Compare that to the surface area option that we have. So the surface area of this device is 115.8 centimeters squared. The ratio we would use for this device is 3 centimeters squared per mil. So that means the same device, using the same chart, the same standard, we would extract in 38.6 mils. You guys see the problem here? With weight, we added almost 500 mils. With surface area, we only added 40 mils, which means the weight ratio would be 12 times more diluted than surface area, okay? What that means is that you'd pass the test probably with weight, and surface area you'd have less of a chance to pass. And so that means everybody wants to run out and do weight because that means you're almost guaranteed to pass. But the FDA has gotten wise to this, and if you've ever submitted weight in the last probably 2 years, you've probably gotten a response that says, please justify why you chose weight and not surface area, okay? Or if you haven't, you will from now on, I guarantee it. So for that reason, we've been using surface area in all our tests. That same approach needs to be applied to the impact of a change of a device. What's the impact to the surface area of that device, that change? And that will help me decide what kind of testing we might need to do or what evaluation would have to be done to evaluate that impact, okay? So this is the impact. We use the same kind of ratio thought that per the standard. If the change is less than 10% of the patient contacting surface area, so if the change of the surface area is less than 10% of the total patient contacting surface area, then the impact to the patient is very small, and we can get by with more an aggressive type of test setup. If the change represents 10 to 50%, we can still be more aggressive than just repeating Biocomp, but it'll take a more test to get to that point. And then, if it's greater than 50%, we are looking at a brand new device at that point, and we really have to evaluate if we need to repeat biocompatibility or we can get by with a limited test scale, but it's a little harder. The main categories you have to consider, though, obviously with any biocompatibility, is what the material is that you're changing to and any processing that's changing. So you look at the surface area impact, what the material is, any processed residuals that could be on the device with the change, and that helps evaluate what kind of need is there, okay? Each change is a little bit different, but I'm going to tell you what the 3 most common ways that we test for the change of the device. These are the 3 tests that we most commonly use to evaluate a change. Cytotoxicity, and we'll go through these tests a little bit in a minute, but cytotoxicity is going to be your best friend and your worst nightmare at the same time, okay? Cytotoxicity historically is the most sensitive biocompatibility test we have. In fact, in the last 13 years at Nelson Labs, if you look at all the failures in biocompatibility, 93% of the time, it's going to fail cytotoxicity, okay? That doesn't mean you're going to fail cytotoxicity 93% of the time. That would be horrible. But what it means is if you're going to fail, 93% of the time, it's going to be the cytotest. That's why it's your worst enemy. But it's your best friend because it's extremely cheap and quick, Okay? At Nelson Labs, it's about $165 to do a non-GLP cytotox test and takes 5 days. Okay? So you got the most sensitive test, but it's the quickest and cheapest. It makes it great for screening, makes it great for screening changes of a medical device. Okay? Another test that we use is FTIR. We'll get into this one too a little bit, but it helps. It's a chemical test that looks at plastics, the bonds between plastics actually, and we give you a comparison between plastics. So if you're changing to a polypropylene or a manufacturer of a polypropylene to a new manufacturer of the same polypropylene, we can run it and actually compare it to see exactly how similar those two polypropylenes are. And then, of course, we have extractable leachable testing, where we can actually look at the impact to a change to the chemistry what's coming off a device. So these are the 3 most common tests that we perform when evaluating the impact of a change of a medical device. And if these tests come back positive, which means good, then a lot of times we can justify out of a lot of the expensive animal tests that may be associated with biocompatibility. So to go in a little bit detail about these 3 tests so you kind of know how we use them to evaluate change, the first one is that Cytotox. Cytotox really is just evaluating the impact of your device on cells. That's what the test looks for. So we grow up cells in these flasks, and then we break these cells apart and put them in these little wells. And then we extract your device, and we put that extract onto the plates to see what happens. If there are toxic chemicals that come off your device into that solution, then they interact with the cells and they kill them. And that's why it's so sensitive, because these cells are just on the bottom of the plate. There's no body system around to protect them. So that's why it's a great screening test, and it's quick and cheap. These are our extraction solutions. We use culture medium for our cytotoxicity tests. To be extracting that culture medium, it's an MEM fluid, which is this pink kind of fluid right here. And we use that to test with. So we put your device in that fluid, we let it sit at 37 degrees, and then we test it. This is the kind of a dashboard for the test, so you kind of get an idea. And by the way, if you want a copy of this presentation, I'm happy to send it out to you. Just give me your business card afterwards, and I'll send you a PDF copy so you don't have to try to write down everything verbatim. But it gives you an idea of turnaround time, sample requirements for cytotox, and some of the problems we usually see with cytotoxicity. Latex is our positive control. So if you have latex in your device, you're going to probably fail cytotox. We most likely see problems with silver, copper, zinc. Those metals are all cytotoxic. It just happens to be some of the dark colors of inks, like blues and blacks, have a lot of copper and zinc in them. So if inks leach off the device, they'll also cause cytotoxicity. In fact, a little bit of a soapbox for inks right now. If you're adding a colorant to a device, please don't. Just don't add a colorant to a device. The FDA has heartburn with colorants right now. The only way I would add a colorant is if it would be beneficial to the functionality of the device, or if my marketing department forces me to add a colorant. But please tell them how much it's going to cost to justify adding that colorant. I can help you give you numbers if they want you to add a colorant, and see if it's really going to be that beneficial in cells to add a colorant. But the FDA right now has a really heartburn with adding colorants. But curing times, short curing times are also things we have problems with. At Nelson, we call it the smell test. You open up a bag and you can smell that chemical smell coming off. If you can smell it, it's coming off in the air, it's going to come off in my extract. Okay? So that means something hasn't cured all the way in your device. So these are some of the things we look for cytotox. Once we get the extract on the cells, then we do a score from 0 to 4. And this scoring criteria is in the standard. A 0 is no cytotoxicity whatsoever. A 1 is less than 20% of the cells have some cytotoxicity. A 3 is between 20 and 50%. A 4 is between 50 and 70%. And anything over 70% is, a 3. Anything over 70% is a 4. And this is also the only biocompatibility test that has an acceptance criteria built into the standard. In the standard, it says 2 or less is considered passing. So you can actually have up to 50% of the cells affected in some way and still pass this test. It just shows you how sensitive this test is. Okay, so I'm going to give you a little bit of video to kind of show you what these cells are supposed to do. Okay, so these cells right here, maybe I am, there he goes. These are my L929 cells that we look for. Maybe I am. Okay, maybe it's not going to work. Okay. Well, these cells grow and divide into this monolayer. Okay. So they'll grow and so this is what they look like when we seed the plates. So you see them starting to stretch and grow, but there's a lot of space in between. Then they'll grow overnight and cover the plate like this. So this is what we want to see. We want to see a complete covering of the plate. We want to see these cells growing and happy. And that's compared to this, which is our positive control, which is a 4. As you can see, there's a pretty big difference between a 0 and a 4. Okay? So there's a lot of blank areas in the cells where nothing's there. What looks like cells are actually the skeleton of the cell left behind, because latex is a fixative, which burns the cells to the bottom of the plate, which is pretty gruesome. So this right here would be a complete destruction. This one down here might be still alive, but not doing very, very well. So this is a 4. This is bad, right? And that's a 0. That's what we want. But the cytotox is great for changes because, like I said, we can evaluate a small change with looking at the impact of a relatively quick and inexpensive test. Okay? So if you have less than that 10% patient contacting change, cytotox is a great way to evaluate that if that change is using materials that are well categorized in the medical device field. The next test that we can use to look at is the FTIR. Okay? So this is what the FTIR does. It basically direct scans only polymers. So if you're changing metals, the FTIR is not going to help you. It's only for polymer materials. And they show the internal bonding or the functional groups. What we can do, though, what's the power of the FTIR, is we can compare polymers to one another. And we can actually compare it to a USP grade polymer too. So let's say you're adding a polypropylene or changing to a polypropylene. We can take your polypropylene, scan it, and then take a USP grade polypropylene, scan it, and give you a comparison, a percent comparison. That's money when you're going to the FDA. You can say, look, we're as good as a USP grade polypropylene or whatever. There's a bunch of different plastics we can use. Or if you have a predicate device that has the same material that's on the market that's shown to be safe, once again, we can do a comparison to be able to give you a percentage. In fact, this is exactly what you look like. Now, if you're not a chemist, this looks like a bunch of mountains and valleys, right? But to a chemist, this is like golden. And what you care about is up in the top there, you see that 95.5. You probably can't see it because it's really small, but you can get the presentation like I said. But this has a 95.51% match between these two. Anything over 95%, we consider a good match. So if you're changing a medical device and you want to be able to, in a polymer, and you want to be able to compare, you can do this, and as long as it's over 95%, that gives you a lot of meat in your justification to be able to show. The last tool that we use to help evaluate changes is chemical characterization. And this is probably the most powerful one, but it's also, out of the three, the most expensive and long. So we probably use this one most likely if you're in that 10 to 50% range, where you don't want to repeat your biocomp, but you're still looking to justify and to help your evaluation. The chemistry can really help you do that without, well, long animal tests and expensive animal tests. Very high level, because I could do a whole 2-hour presentation on chemical characterization. I'm going to kind of talk to you a little bit about what chemical characterization means. Really what we're trying to do is get a fingerprint of what compounds come off that device. So we take your device, we extract it in certain solutions like water and hexane or isopropyl alcohol, and we use those alcohol or the solutions and put it on chemistry to evaluate what compounds are coming off your device. That really is a fingerprint. We know exactly what is coming off your device into the body. Now we can take your change of your device, your new device, run the exact same extractions and chemistry tests, and we can compare the number and types of compounds that come off your original device and your change to evaluate the impact. And most of the time, that does not change very often, especially if you're in that 10 to 50% realm. Once you get over 50%, then we start having issues. And if you do have changes on the extraction, then we can evaluate the impact of those changes. We can look at what compound might be higher or in higher amounts or a new compound, and we can evaluate the safety without going to the animal tests. So if you're within that 10%, maybe just an FTIR and cytotox would be sufficient based on what material you're changing. If you're in that 10 to 50%, we might have to go into the chemistry to evaluate more. And then if over 50%, we might have to add some of the animal tests in, depending on what the change is and what the materials are. Some of the things we look for, I put it down here, these are the things that would come off a device that we would see in an extract, just some of the examples. These are the testing scheme we look for an extractable leachable test. We look for volatile and semi-volatile compounds. To give you an idea, volatile compounds are just ones that escape really quickly. They're small molecular weight, so alcohol sitting on a desk would evaporate. That's a volatile compound. Semi-volatile is just a little bit slower, so probably me in a race. And then me, probably the non-volatile compounds, those are the ones that aren't going anywhere, right? So those are the ones that are staying. They're like metals, some plasticizers. Those things are non-volatile compounds. But we look for all three areas of those compounds to try to see what fingerprint comes off your device, okay? So that helps us evaluate that impact of that change. Once again, I talked about colorants. Let's just not put colorants in, right? We decided that. No more colorants. So in conclusion, I wanted to sum up. The impact of the change really is based off the percentage of the surface area that's changing in the device. The patient contacting surface area. If it's less than 10%, we usually can get by with doing a cytotoxicity and an FTIR. If it's more than 10%, we might have to add some more chemistry, maybe a couple of other animal tests. Anything over 50%, then we have to be a little bit more aggressive in our testing scheme. But depending on what the materials you're changing to or the type of change you're performing, we can be very aggressive in this type of assessment. In fact, in the last 2 or 3 years, we've been in the high 98% rate with submitting change assessments to the FDA and accepting them for new changes of devices. Because really you have a predicate on the market that you can compare to, and that's what the FDA is looking for. And so it's a really easy and safe way to do an evaluation. Any final questions? Yes? So if I understand your question, you have the same material, right? It's just a different manufacturer of that material? Okay, open cell or closed cell foam. So any change could have an impact in biocompatibility, but that type of change would be one of those that we could evaluate through an FTIR or in a cytotoxicity and not have to do all the biocompatibility tests. So they could talk about a closed cell foam may not leach as much as an open cell foam, for example. So the FDA may have some concerns. The best thing to do is not to submit to the FDA and cross your fingers that they're going to accept it, but come out with a proactive plan. Say, this is my change, this is how I evaluated my biocompatibility, and then they are more likely to accept it than just crossing your fingers and hope they didn't want to see biocompatibility. And the FTIR and cytotox are very cheap and quick, so it's a good way to evaluate it. Good question. Any other questions? Okay, if you have did you have one? Sorry. Very good question. I get this question. If you guys didn't hear it, I get this question all the time. So if you have an aged product, right, does the biocompatibility change? Like if you have a 3-year shelf life, and does the biocomp change at time 0 in 3 years or 5 years, whatever it is? It depends. And I hate saying that answer, but it does. Most of the time, biocomp gets better over time. I can take my latex and I can put it on my the counter of my lab and leave it there for a year. And as long as I don't cut it and test it, it would pass. Because a lot of those compounds that we're looking for are volatilized off, right? But if the material degrades over time, then we have issues. So it's all depending on the material. Most materials, though, are better after time. I do have companies who want to evaluate biocompatibility at the end of aging. They just throw an extra sample into the accelerated aging, and we run a cytotox at the end of it to evaluate it. But as an ISO rule, the brand new device off the line is worst case, unless it degrades, unless it's a degradable material. That's when we want to do biocompatibility. It's a very good question, though. Yes? Okay, another good question. If you didn't hear that, how does changing of sterilization impact biocompatibility? I'm going to answer, she said just E-beam to gamma, and I'll answer that. But just so you know, in Nelson Labs, there's an EO department and a radiation department, and they always fight at which one's better. Okay, so they always yell at each other about which one's better. I don't care. I'm neutral. I'm Sweden. But I will tell you, EO is much better for biocompatibility than gamma is. The reason why is because the only thing you have to worry about with EO is residuals, and you do residuals, and so you have irritation already in your residuals. Where gamma changes cross-linkers, especially in polymers, and can change the biocompatibility. I've seen devices fail before gamma and pass afterwards, and I've seen devices pass before and fail afterwards. So I can't tell you if gamma is going to be good or bad. But by changing to gamma to E-beam or E-beam to gamma, it all depends about the dose of energy, because they're both just energy. So if your dose is the same, then you don't have to worry about biocompatibility. But if you're changing the dose, then we have to evaluate it. But FTIR and Cytotoxicity a lot of times have a good evaluation with that kind of thing. If you change from gamma to EO or EO to gamma, the chemical characterization has been invaluable with that. We've helped many companies change between gamma and EO by looking at the material characterization of the gamma and the material characterization of the EO and comparing the two to see if there's any differences. Did that answer your question? That was probably a really long answer to a short question, but that's what you get. So, any other questions? Okay, if you have specific questions, like I see changes, that's mostly what I see. So if you have a change that you just want to run past me, what we've done in the past, I can probably help you. We're at booth 420, so if you want to stop by. But thanks for your time, I really appreciate it.